Selection of drug candidates. Introduction. Selection of drug candidates for controlled release dosage forms is vitally important. Drugs that undergo or may undergo hepatic first-pass metabolism should not be formulated in a controlled release dosage form, because this will reduce their absorption rate, unless justified by the fact that this process gives rise to an active metabolite. Conversely, a little medical justification exists for the drugs having a long biological half-life, that is, greater than 12 hours, and are to be formulated in a controlled release formulation, since these drugs are long-acting. Unless the development of a controlled release formulation offers some convenience to the patient and improves patient compliance. Ideal properties of a drug suitable for sustained release drug delivery system, SRDDS. 1. It should get effectively absorbed on oral administration and should remain stable in gastrointestinal fluid. 2. It should have a short half-life, 2 to 4 hours, e.g., captopril, salbutamol sulfate, etc. 3. Its dose should not be less than 0.5 gm and its maximum dose for designing SRDDS should be 1.0 gm. Example, metronidazole. 4. It should have a wide therapeutic range so that any variation in drug release does not result in concentration beyond the minimum toxic levels. Compounds that are unsuitable for controlled release drug delivery system, CRDDS. Certain drug candidates are not considered suitable to be formulated in controlled release dosage forms owing to some of their properties. For example, drugs with an elimination half-life less than 2 hours, or drugs having large doses to administer within the body, or drugs having a half-life greater than 8 hours are not required to be formulated as controlled release dosage form. Thus, drugs with the following properties are considered inappropriate for designing CRDDS. 1. Drugs with a short and long half-life. 2. Drugs undergoing hepatic first-pass metabolism. 3. Drugs with a low solubility. And 4. Drugs requiring to be administered in a large number of doses. Properties of drugs relevant to controlled release formulations. The release rate of drugs from controlled drug delivery system are affected by the following two major factors. 1. Physiochemical factors, and 2. Biological factors. Physiochemical properties. The following physiochemical properties of the drugs should be considered while selecting drug candidates for controlled and sustained release dosage forms. 1. Molecular size and diffusivity. Drugs formulated in sustained release dosage forms should diffuse through various biological membranes and also through a rate-controlling membrane or matrix. Diffusivity is a drug's ability to pass through membranes, and it is a function of its molecular size or weight. Molecular size of the diffusing species has a significant influence on the diffusivity value, D, of polymers. Thus, the value of D is related to the size and shape of the cavities and of the drugs. The diffusion coefficient values of drugs having intermediate molecular weight, 150 to 400, through flexible polymers, range from 10 to the power of 6 to 10 to the power of 9 cm squared, sec, and the values in 10 to the power of 8 order are the most common. The diffusion coefficient values of drugs having molecular weight greater than 500 are so small, i.e., less than 10 to 12 square centimeters, sec, that their quantification is difficult. Thus, drugs having high molecular weight should display very slow release kinetics in sustained release devices where diffusion through polymeric membrane or matrix is the release mechanism. 2. Aqueous solubility. Drugs are mostly weak acids or weak bases. Drugs are formulated into sustained release dosage forms with very difficulty if their water solubility is low. While it is difficult to delay the dissolution rate of drugs having a high water solubility and rapid dissolution rate. Drugs having a high water solubility readily dissolve in water or gastrointestinal fluid, are released in a burst, are quickly absorbed, and thus their concentration in blood is more than the drugs having low water solubility. Incorporating a highly water-soluble drug in the dosage form and retarding the drug release is a difficult task, especially when the drug dose is high. The pH-dependent solubility in physiological pH range is another problem for sustained-release dosage forms due to the variation in pH of gastrointestinal tract and in dissolution rate. 3. pH and pKa. Highly ionized drugs are poor candidates for oral sustained-release formulations. Unionized drugs undergo appropriate absorption, while the ionized drugs undergo negligible permeation as their absorption rate is 3-4 times less than that of the unionized drugs. An acidic drug with pH-sensitive ionization has a pKa range around 3.0 to 7.5, while a basic drug with pH-sensitive ionization has a pKa range around 7.0 to 11.0. And such drugs are ideal for optimum positive absorption. The drug should be unionized at the site to an extent 0.1 to 5%. 4. Partition coefficient. It is the fraction of drug in an oil phase to that in an adjacent aqueous phase. Partition coefficient, K, influences drug permeation across the biological membranes, and also drug diffusion across the rate-controlling membrane or matrix between the drug administration time and drug elimination time. The drug should diffuse through various biological membranes that act as lipid-like barriers. Apparent oil or water partition coefficient is the major evaluation criterion of the drug's membrane permeability, that is, its ability to penetrate these lipid membranes, and is expressed as K equals Ca divided by Cw, where Ca equals equilibrium concentration of all forms of drug in an organic phase at equilibrium. Cw equals equilibrium concentration of all forms of drug in an aqueous phase. Relationship between drug action and partition coefficient log drug activity log K. Generally, the drugs having a high K value are highly oil-soluble and readily partition into the membranes. Hunch correlation defines the relationship between tissue permeation and partition coefficient for the drug. This correlation describes a parabolic relationship between the logarithms of its partition coefficient phi, permeability, log P, expressed lipophilicity, and molecular size of the drug are the three drug properties that influence the drug's permeability for passive transport across intestinal epithelium. Another important factor that determines the drug permeability is the polarity of drug, which is measured by the number of H bond acceptors and H bond donors on the drug molecule. 6. Mechanism and site of absorption. Drugs to be absorbed by carrier-mediated transport are not suitable to be formulated as controlled-release systems. For example, vitamin B2, a water-soluble vitamin, is required for several metabolic reactions and for preventing medical problems, especially hematopoietic conditions and spinal cord-related neuropathics. Vitamin B12 ingested in its free or non-protein-bound form will bind to a carrier protein, namely R binders or trans-cobalamin I, that is secreted by the salivary glands in oropharynx and by the gastric mucosal cells in stomach. 7. Drug stability. Loss of drug through acid hydrolysis and/or metabolism in gastrointestinal tract is an important factor for oral dosage forms. A solid drug degrades at a much slower rate than a drug in suspension or solution form. Thus, the relative bioavailability of a drug that is unstable in stomach can be improved. The most appropriate controlling unit for the drugs that are unstable in intestine is the one that releases its contents only in the stomach. Therefore, drugs with stability problems in any particular part of the GIT are considered as poor candidates to be incorporated into controlled release systems that deliver their content uniformly throughout the length of GIT. In controlled drug delivery systems, The drugs are protected from enzymatic degradation as they are incorporated into a polymeric matrix. Thus, these systems are beneficial for highly unstable drugs.